Great. Welcome, everyone. I'm just so excited to see everyone. It's been a while, and I'd like to thank everyone to be here. This is our inaugural Cold Spring Harbor Cell and Membrane Fusion meeting. And like Andy, I'd like to also thank the Cold Spring Harbor Laboratory for providing us this beautiful home for our meeting, for our community. And hopefully this is going to become a long-lasting tradition in years to come. I'd also like to have a special thank to our co-organizer, Andy, who has done the lion's share of work in organizing this meeting with her energy, and you can see the energy is like exuberating, and all the careful planning. I think she deserves a big round of applause. And she was like, oh, you and Mark have to let me know, tell people what I have missed in my intro, and being a superb organizer, she hasn't missed anything. So I am going to start with the intro of our keynote speaker, okay? It is my wonderful pleasure to introduce our first keynote speaker, Jennifer Lippincott Schwartz. And Jennifer is a senior group leader at the Janelia Research Campus on the HHMI, of the HHMI Howard Hughes Medical Institute. Her research uses live cell imaging approaches to analyze the spatial temporal behavior and dynamic interactions of molecules in cells. Jennifer has a rather unusual start on her academic career. She majored in philosophy and psychology in college and spent some time teaching science and math in Africa afterwards. And when she returned to the States, she decided to pursue her passion in biology and worked with Philip Hannawald's lab at Stanford, studying enzymes involved in DNA repair. And as she studied the roles of individual proteins, she was curious about how the cell as a whole would function. And that curiosity led her to Doug Fembro's lab at Johns Hopkins University, where she studied lysosomes using antibodies and microscopy, and reviewed the dynamic nature of lysosomes. And in her postdoctoral work with Richard Klausner at the NIH, she studied the secretory pathway and continued to debunk popular notions of static, unchanging organelles. And with a strong motivation to visualize the dynamics of proteins in cells, Jennifer's own lab at NIH pioneered the use of GFP to tag proteins and visualize membrane trafficking between the ER and the Golgi in live cells, and forever changed the conventional static view of protein distribution and function in cells. She refined the FRAP method, which a lot of people are using, and developed a flip method to study cellular dynamics. And in 2002, her lab developed the photoactivatable GFP, which provided the critical tool for Eric Betzik and Harold Hess to invent the photoactivation localization microscopy, known as PALM, which was the first super-resolution imaging technology, which a lot of us have been using. And Jennifer's lab continued to develop new tools to look at the inner makings of cells. Her work touched many, many areas of cell biology, just to name a few: protein transport, the cytoskeleton, organelle dynamics, organelle contact sites, lipid droplets, cell polarity, and cell fusion. That's why she's here today. And every time I see Jennifer's talk, I would be amazed by the beautiful images and videos of cells captured by the new tools, be it new probes or microscopy developed in her lab. And I look forward to seeing more of those today. Jennifer is widely recognized for her seminal contributions to cell biology. She was the president of the American Society for Cell Biology and was awarded the E.B. Wilson Medal, which is the highest honor of ASCB. She's an elected member of the National Academy of Sciences, the National Academy of Medicine, the American Academy of Arts and Sciences, and the European Molecular Biology Organization. And we're so extremely excited and honored to have you here to give us the keynote talk today, Jennifer. Welcome.
Thank you so much. I want to start by thanking Andy, Mark, and Elizabeth for this invitation. I've never been to a cell-cell fusion meeting before, so I'm very excited. And to answer Andy's question, which is, have you ever have you ever worked in cell-cell fusion? I will answer yes, a little. Do you dream about cell-cell fusion? And I'd say yes, a lot. So today I'm going to be talking about work that we've done that is sort of in that dreamlike world where we're trying to understand what exactly is cell-cell fusion doing for sort of the fundamental underpinnings of the cell. Because when cells fuse to create syncytia, there's really magic happening in these syncytia. So these are just a few examples of syncytial systems that arise as a result of cell-cell fusion, including, as everyone knows, skeletal muscle formation, where cells fuse into this myotube, which then differentiates into myofiber. Bone development requires these osteoblasts, which are huge multinucleated syncytial cells that are resorbing the bone. In immune responses, macrophages will differentiate into giant cells that have many nuclei, making this giant cell very effective at attacking target cells. And finally, probably the most important for humans is placenta, the placental development. Placenta is one big syncytia, and in fact it was our interest in placenta that originally got my lab interested in cell-cell fusion. Because in this system you have trophoblast cells that, once the blastocyst is beginning to implant, will fuse into this syncytial trophoblastic layer that will create the placenta. And in so doing, this layer here takes on characteristics that enable the placenta to serve many, many roles that are beyond what a single cell can do. This placenta is acting as the lung for this embryo. It's acting as the liver. It's acting as a blood flux. So many new properties are coming to this syncytial system, and that's what really got me quite interested in what is happening to this system when it moves from one cell to multicellular. So I want to just sort of highlight, make sure we're all on the same page in terms of what this process is about. We know it's tightly controlled. Not all cells in your body can undergo cell-cell fusion or normally undergo cell-cell fusion. I'm going to show you, yes, you can make pretty much any cell undergo cell-cell fusion. All you really need is for these cells to express fusogens. But the way that syncytial formation and cell-cell fusion is being regulated developmentally is through restricted expression of specific types of fusogens. And I'm sure many of you know it in the audience, but if you don't, in the case of placenta, the fusogen is a retrovirus that was taken up into the animal host. So basically, it's only because of retroviral infection that placental animals came into existence. So it just underscores the role of these fusogens in enabling these complex cell-cell fusion systems to emerge evolutionarily. Now, one of the things that happens when you have cell-cell fusion is that there is a halt in cell proliferation. There's also transcriptional reprogramming, and new traits emerge in this syncytium. So what I'm going to focus on today is work that we've done addressing the question is, how are these changes, these new transcriptional changes and this halt in proliferation actually arising? And the question we were in particular interested in is, could cell-cell fusion itself contribute to these programmatic changes that generate the new syncytial cellular state? Or is it pre-dependent on the cells that are expressing the particular type of fusogen in their particular environment. That's really critical in terms of the programmatic changes. So how do we address this question? And it's simple. Let's try to fuse any cell in the cell and look to see what happens to those cells. Do they undergo any kind of programmatic change as a consequence of being artificially driven to be fused? And I remember when we first decided to do this experiment, I was like really excited because I said, what's going to happen when two cells fuse? Are they going to change? They're going to have two nuclei or four nuclei. Are they going to change fundamentally? And if so, what explains that? So anyway, what we're going to do is we're going to take a cell. In fact, we can do this with any cells that we transfect with VSVG, which is a fusogen. VSVG in the presence of LDL receptor. If LDL receptor is on one cell and VSVG is on another, when those cells come together in a low pH environment, they will fuse. And that's our protocol here. We're going to take a VSVG infected cells, and we're going to use these SUM159 cells, which is a fibroblast cell line that has LDL receptor on its surface. We're then going to do 5 to 10 seconds of isotonic buffer at pH 5.5, and then watch the cells fuse. And this is what we see. So these are 3 cells here. They're expressing a cell surface protein, GPI, GFP, and we're just going to pipette in this low pH buffer and then bring it back up to normal pH and watch what happens. And what you can see is that these cells undergo very fast fusion. And you can actually see, this is lattice light sheet imaging, so we're watching, this is looking at the top of the cells as they're fusing, this is looking at the sides of the cell as they're fusing, and you can see that the initial fusion is happening right here, and then very quickly the fusion pore, some type of fusion pore is expanding, and you end up with a single cell made up of several nuclei. Now, if we look at how quickly cytoplasm mixes in this system, this is what we see. So here we're expressing a nuclear tag in yellow here, cytoplasmic marker is in gray, and you can see how fast that cytoplasmic marker will mix within this population of cells that are undergoing low pH induced cell-cell fusion. And this is just quantifying that mixing phenomenon. Here's a donor cell, here's the receiver cell. You can see how fast the donor cell's losing its cytoplasmic signal because it's being taken up and mixing with the receiver cell. And this is just a quantification of that whole process. So soon as cells fuse, you see incredible mixing of the cytoplasm. Nothing's holding the cytoplasm back. Well, what about cytoskeletal components? What about actin and cytoskeleton and microtubules? How quickly do they start intercommunicating between these two different cells when we induce this cell-cell fusion? And you can see that it's pretty fast. I mean, the actin filaments very quickly intermix. In the case of the microtubules, you can see that MTOCs are drawn together into a close approximation with each other. The nuclei also are drawn to, the nuclei do not fuse, but they come in close proximity to each other as a consequence of these centrioles being drawn together. We can also see that organelles, in this case mitochondria, which we're tracking, this is the mitoemerald is in one cell. We have 2 different colors of mitochondria here that are being mixed as these cells are engaged in fusion, and we're just tracking the motion of these microtubules, of these mitochondria, and you can see that the mitochondria don't they ultimately mix, but it takes much longer for them to mix because they're moving along microtubules, so they're just following the microtubule tracks. I didn't show how an organelle like the ER mixes. I know Elizabeth may be interested in that, but the ER mixes also extremely quickly. ER tubules will fuse with the tubules of ER in the neighboring cell, and you'll get very quick exchange of proteins. So basically, there's a sequence of cytoplasmic and organelle intermixing that you can actually put on a timeline when you see cell-cell fusion. So the cytoplasmic mixing happens within a minute or less. Then we see mixing of ER. So here's an example of the mixing of endoplasmic reticulum within this system. That takes about 3 minutes to start getting exchange of ER resident proteins. They start diffusing very quickly throughout the whole ER of the neighboring cell. The mitochondria take a much longer time because they are not fusing with each other. They're just moving as a whole organelle from one cytoplasm into the other. And then finally, the nuclei cluster together. Importantly, they do not fuse. But they come together in a more centralized location. Okay, so that's cool. So that means there's some active events that are drawing the cytoplasm of the cells that are undergoing fusion together. And it's nice to sort of see how that process unfolds in time. Well, one of the things that we could see as well when we started looking into this is that within a very short period of time after cell-cell fusion, we see cell cycle arrest. So we're seeing that 95% of our cells don't divide after cell-cell fusion, and they start expressing this protein P21, which is a cell cycle arrest regulator. So here are cells that haven't yet fused. Here's the syncytium. You can see most of the nuclei in that syncytium are expressing this cell cycle arrest marker. Here are two cells that are poised to fuse with this guy, which is full of the P21 marked nuclei. So basically, there's a huge difference in terms of the extent of P21 positive nuclei, indicating that as the cells, once they fuse, they go into a state of not dividing. Well, what happens to the cells that don't go into this differentiation state? And why would the cells want to go into a differentiation state? Many years ago, we actually looked at this process in a project at Woods Hole. This was one of the students, Olga Alfonso, who used a lattice light sheet microscope to look at, she purposely got cells into sort of a late stage of the cell cycle where many of the cells were going into mitosis. And then she induced cell-cell fusion. And so when she did that, she started seeing cells that are in mitosis, clearly in mitosis, fusing with cells that are not in mitosis. And we can see that based on this nuclear stain here. You can see this is an interphase cell. This is a cell that's started in mitosis. This is a mitotic cell. That's a mitotic cell. These are interphase cells. That's a mitotic cell. Okay, I'm going to play this movie, and you're going to see that as soon as these cells start fusing with this syncytium, these nuclei go instantly into mitosis. It's really remarkable. So all of the nuclei go into mitosis very, very quickly, and then the whole thing blows apart. Because the cells are trying to divide, and there are too many nuclei undergoing division, and the cell blows itself apart. We see this time and time again. Here's another example. Here's a mitotic cell. Here's a mitotic cell. These are interphase cells. These guys have not fused yet, but they're poised to fuse with this syncytium here, or this thing right here, which is a fused system. It's a fused system that is got nuclei that are in mitosis, which means that the cytoplasm of this system right here is full of mitotic drivers. And what's going to happen is those mitotic drivers are going to instantly put these guys into mitosis. And you can see this again. You can see instantly, as soon as you had fusion, the nuclei of the incoming cell, when it experiences a mitotic cytoplasm, it will drive itself into mitosis. And once you have too many mitotic nuclei in the syncytia and they're trying to divide, they blow the whole system up. So that's probably one of the reasons why when cells fuse, they do not want to be in a proliferative state. They need to turn on a way to shut down proliferation and go into differentiation. Okay, so one of the things that so our data is definitely supporting that. Our data is supporting in our model system for cell-cell fusion using VSVG, what we're seeing is that the active cell-cell fusion alone is propelling the syncytium toward a differentiation state. And so in order to look at that more carefully, basically, Daniel Feliciana, who is the driver of all the work that I'm talking about today, he was a postdoc in the lab, but he now runs his own lab at Janelia. He did RNA-seq of control and fused cells. He then examined basically the pattern of genes that were turned on, up or down, as the cells moved into this fused state. And so basically, there were about 2,500 differential genes when you compare fused versus non-fused cells, some 294 cells that were either not inducing to fuse or inducing to fuse. And we're seeing a huge number of proliferation genes being downregulated and differentiating genes being upregulated. We can see use the RNA for the P21 actually going up in our fused cell system, which is very encouraging. But we also see many proteins or genes that are downstream of YAP1 being impacted by this. So YAP1, when it is expressed, it is one of the major drivers. It's a transcriptional co-activator that is a driver of proliferation. And so obviously you would think that YAP1 would not want to be active in the syncytium. Now, suggesting that, we're seeing that many YAP1 downstream genes are actually downregulated in the differentiated RNA-seq data. So that led us to start looking at YAP localization and cell fate determination vis-a-vis YAP in our fused versus non-fused state. So just to sort of get everybody on the same page, when YAP is in the nucleus, it's promoting cell proliferation. And that's where it's located in stem cells or cancer cells, tumor cells, I should say. And that's when the hippo pathway is off. When the hippo pathway is on, and that hippo pathway is what draws YAP to the periphery of the cell, where it's hanging out with cytoskeletal stuff at the periphery of the cell. But basically, conditions that turn on the hippo pathway lead to cytoplasmic YAP, and YAP moving out of the nucleus, and that promotes cell differentiation because you no longer have YAP to turn on all those proliferation genes. And this is what you see typically in differentiated cells. So we decided to look at what happens to YAP localization in our cell-cell fusion assay. And this is what we see. So prior to cell-cell fusion, before we've done our low pH shift, we can see using an antibody YAP that there's a lot of it in the nucleus. This is our control right here. Within 5 minutes, however, after doing our low pH shift to drive cell-cell fusion, so you can see these cells are in the process of fusing, we start seeing, so now the cells are fusing here. Within 15 minutes, when the cells have created this syncytia, we now lose the YAP localization in the nucleus. And by 60 minutes, we see depletion, dramatic depletion of YAP from the nucleus. And that's just plotted here. So that was pretty exciting. And basically, this is in the VSVG artificial system. But we can also see this. I'm sorry. This is just quantifying if you come in with Western blot to look at YAP, we can look at the phosphorylated form of YAP as well as the total YAP. And you can see phosphorylated YAP, which is cytoplasmic YAP, is dramatically increasing post-fusion. This is all happening in this VSVG-induced cell fusion system. But we can also see the same thing happening with trophoblasts that are induced to undergo cell-cell fusion as a result of a hormonal trigger. And so what you can see here is before we've triggered cell-cell fusion among these trophoblasts, YAP is in the nucleus, and it moves out into the cytoplasm, and the nucleus is now depleted of YAP as a consequence of that. We can also see this in a mouse embryo. If you try to look at the placenta here, you can see that the cells that are in this placenta you can see empty nuclei vis-a-vis the YAP signal. So how might cell-cell fusion drive YAP to this cytoplasmic localization? There are many known YAP regulatory cues that induce changes in nucleus cytoplasmic ratio of YAP. One is metabolic stress. If cells are starved, YAP cells don't want to divide if they're starved, and YAP moves out of the nucleus into the cytoplasm. Mechanical stress is another example. If cells are mechanically stressed, you can see YAP moving out into the cytoplasm, and the same with osmolarity changes. You can actually drive YAP into the nucleus if you take water out of cells. So we wanted to test whether there's any biophysical parameters that we're seeing in our model system using VSVG to induce cell-cell fusion that might impact these cues. And one idea we had is cell surface area. What's happening to the surface area of the cells that are undergoing cell-cell fusion? Because they could absolutely impact mechanical stress. There could also be some sort of osmotic stress if there's also a change in cell-cell volume when these cells fuse. So let me just sort of do a very simple analogy here. You all know that if you take two objects like these two squares and you fuse them into one, there's excess surface area that needs to be removed when you combine these two volumes. So when you're combining volumes and you're keeping the volumes constant, you're going to have excess surface area. So we decided to see what is the situation in terms of the volume and the surface area of the cells that we're looking at when they undergo cell-cell fusion, because we felt that these changes might impact the downstream effects on YAP and potentially other things happening at the transcriptional level in these cells that have fused. Because we got to understand, why the heck do you have these dramatic changes when these cells fuse. It's the same cell. It's within minutes. Something's happening big. So what is it? So let's look at the surface area. So this is a plot. We're color coding the surface area of this cell. This is before fusion. This is the two cells in contact with each other. We're going to now drop in the low pH, and we're going to watch what happens. And this is just plotting the change in surface area as a function of time. You can see it's dropping. So the surface area is undergoing a fairly significant drop in surface area as a function of cell-cell fusion. In fact, you could see the long filopodia sort of being absorbed into the system. So here's just some quantification. This is pre-fusion. This is 30 minutes after fusion. If we look at the surface area distribution of these guys before and after, you can see a big shift. Post-fusion, you have a decreased surface area. You have in the case of volume, the volume of these two cells before and the volume of these two cells after fusion are pretty close. There may be a slight drop, but it's pretty close. When we did this experiment, I was rooting for volume change. But we didn't get a big volume change. What we got was a very big surface area change. Now, it sort of makes sense that you don't want to change the volume of these two. You don't want to dramatically alter the volume of these two cells when they come together, because changing the volume of a cell is hugely impactful for that cell. It's not so hard to change the surface area because all you have to do is either shed that membrane or take it up through endocytosis. And so we were interested in knowing, okay, well, what's mediating that surface area reduction during cell-cell fusion? So immediately, Daniel, what he did was he expressed a membrane marker, DID, which you can see here in white. He also has a BFP dye, which is letting you look at the cytoplasm. So you're going to see these cells fuse, and you'll see this blue signal move into all of these cells. And that tells you when the cell-cell fusion is happening. And I want you to pay attention to what's happening to these little dots here, which represent endocytic structures. And when we do this, we see a huge increase in the amount of endocytosis occurring in this system. So that raises the question of, well, what kind of endocytosis might be mediating this? Is it basically caveolin? Is it class 1-mediated endocytosis? Is it class 1-independent endocytosis, macropinocytosis? Basically, we focused in on this class 1-mediated pathway because there's prior work in the field suggesting that class 1-mediated endocytosis can take up membrane from the cell surface when that cell surface is loose. And in fact, during mitosis, when the cell rounds up to create a mitotic cell, you have a lot of endocytosis that's mediated by class 1. So basically, we're going to look at whether class 1-mediated endocytosis is regulating this surface area decrease. And so basically what Daniel did was he looked at class 1 and AP2, which is an adapter for class 1. He looked at their density on the plasma membrane after cell-cell fusion, and you can see that there was a significant increase in the number of these punctae under cell-cell fusion. And this is just looking at it in live cells. You can see a big increase in the endocytic activity. This is quantifying it. This is AP2 spot density. This is with cell-cell fusion. This is with nothing. So basically, what Daniel measured was about a 1.75-fold increase in endocytic activity under cell-cell fusion. We can block this with pit stop, which inhibits the activity of classroom. This is just showing how we can basically, with pit stop, we bring basically the amount of endocytic structures to what we see under normal conditions. So that raises the question. So when the cells are fusing, they're diminishing they have excess surface area initially, and that excess surface area appears to be being drawn in through endocytic pathways. So the question was, is this endocytic activity at all connected to what we're seeing with respect to YAP in the cytoplasm? Remember, we see YAP move out into the cytoplasm under cell-cell fusion pretty early, around the same time that we're seeing this boosted endocytosis. Could they be connected to each other in some way? Well, we looked at that by looking at what happens to YAP redistribution when we block the endocytic path, this endocytic uptake of the surface area. And sure enough, we could interfere with the ability of YAP to be depleted from the nucleus when we inhibited using 3 different endocytic inhibitors. So here's a normal time course of YAP depletion from the nucleus. And when we block endocytosis, at the time that we're driving cell-cell fusion, we don't get the loss of YAP from the nucleus. So this is suggesting that endocytosis is directly affecting signaling to YAP. So how in the world is that happening? Okay, well, let's think about YAP. I mentioned YAP is sensing nutrient stress. One way to get YAP out of the nucleus is to starve cells. So is it possible that when we have this increased endocytosis, we actually are drawing in transporters, like maybe glucose transporters, that actually are critical for the cell to be able to take in glucose. And without them, you're going to have a nutrient stress situation. So to test that, we looked at GLUT1 surface expression. GLUT1 is a glucose transporter. And what we see is that its level of expression on the surface actually decreases upon cell-cell fusion. And furthermore, we can, this is just showing another example of that. We're looking at amino acid transporters, which we don't see any change in their distribution on the cell surface. But we're seeing specifically a decrease in the level of glucose transporters when we have this boosted endocytosis under cell-cell fusion. Now, we think that the glucose transporters are being drawn in because they normally cycle to and from the plasma membrane. So any kind of increase in endocytosis is going to shift their equilibrium towards the inside of the cell. These amino acid transporters don't necessarily undergo this constitutive recycling, and so they're not really affected too much. But the increase in those glucose transporters into the cell upon cell-cell fusion as a consequence of the increased endocytosis, which is occurring to get rid of that excess surface area, should lead to potential glucose depletion from cells. And so to test that, we looked at glucose levels and whether they change upon cell-cell fusion. And for this, we made use of a glucose sensor developed by Lauren Luger at Janelia, which basically, in the presence of glucose, it shifts its spectrum. And so you can see a signal. So here is the signal. This is a color lookup table, and I'm sorry I don't have that the lookup table, but basically we've tuned that lookup table such that when you see blue, it's low glucose, and as you move to green and red, it's high glucose. So in a non-fused cell, what you can see is over the course of 30 minutes of imaging, not much change in the overall distribution of glucose within the cell. This is in contrast to when we drive cell-cell fusion. So at 0 time, we don't have much difference than what we see in a non-fused cell, but as soon as we start, we fuse the cells here, and now when we start looking at the glucose levels in the cell, we see that they drop pretty dramatically, pretty quickly. So within 5 to 10 minutes, we're going down to a nadir of glucose level within the cell. And then slowly over the course of the next 30, 40, 60 minutes, the glucose levels come back slowly to normal levels. Now, interestingly, if we look using luminescence assays at ATP levels in these cells, we also see a drop in ATP that's corresponding to the drop in glucose, which makes sense because when you don't have glucose, you can't drive glycolysis, and glycolysis is an important ATP producer in cells. So that led us to then basically focus in on whether this low ATP could be triggering a key sensor of ATP energy levels in cells, which is AMPK. And AMPK talks to YAP. So that's why we're looking at this AMPK. And what you can see is AMPK upon cell-cell fusion, the phosphorylated form goes up. You can see that here. This is untreated cells, pre-fusion, 5-minute fusion, and then 15 minutes and 16 minutes. The phosphorylated form is, you can see, it's low here, it goes up, it's up, it's up. If we use this compound C, which blocks AMPK activity, it's an inhibitor of AMPK phosphorylation, you do not see that rise in AMPK. So what we see is that inhibiting AMPK results in YAP nuclear retention. So basically, if you do not allow AMPK to get activated, we actually don't get you can't get YAP out of the nucleus. So what this is telling us is that this redistribution of YAP in and out of the nucleus, from the nucleus into the cytoplasm, is connected to the energetic state of the cell that's being altered transiently as a consequence of the cell-cell fusion. And that cell-cell fusion, as I mentioned, is boosting endocytosis, which is driving the glucose transports inside the cell, which lowers glycolysis, drops your ATP, and triggers this very quick redistribution of YAP out of the nucleus, which then stops proliferation, drives the cell into a differentiated state. So I want to sum up what our data is suggesting, and I should say we're using a very simple system to drive cell-cell fusion. We did this on purpose. First, I was betting nothing was going to happen when we first started. But then we saw some major things happening. And when we started dissecting these major things, we started realizing that this is conserved for other types of cell-cell fusion systems that are happening throughout these developmental systems. So basically, our data is suggesting that a general principle of cell-cell fusion is a structural to transcriptional signaling pathway that's mediated by an endocytosis AMPK YAP axis that's triggered upon cell-cell fusion. What this is doing is linking membrane remodeling in the boosted endocytosis and cellular bioenergetics, starvation when you downregulate GLUT4, to transcriptional reprogramming, YAP moving out of the nucleus in response to cell fusion. Now, AMPK, which is the ATP sensor in the cell, is playing a central role in this whole process because It's what's sensing the transient reduction in cytoplasmic glucose and ATP that's caused by the modification in the plasma membrane landscape through increased clathrin-mediated endocytosis that's due to the attempt by this syncytium to reduce its surface area to conserve its volume. So in tissues, we believe that this structural transcriptional signaling system is likely synergizing with additional environmental cues to accomplish what these syncytia have, which are very specific differentiated states. So with that, I want to end and thank the people involved in this. My biggest thanks go to Daniel Feliciana, who, as a postdoc in the lab, led all of this work, but he was very much helped by other members of my lab. Thank you so much. I don't know how much time we have, but
Right.
I'd love to answer any questions.
Time for some questions. Well, Andy was saying a trainee first question. Okay. Yeah.
Hi. Okay.
Oh, back there? Okay.
So I'm first. Hi, thank you for the talk. So I wonder, like, when you're saying after you use the PhD-induced cell fusion, that the YAP got redistributed and it become a differentiated state, what extra function does the syncytia get? Like, for example, like, how is it doing better deal with stress, or do better with like something like tissue repair, DNA repair, something like that?
Yeah. We have all the data for what genes are up, what genes are down. We don't, this system may be, we haven't tested specifically whether the system, the artificial system that we have, is more resistant to stressors or less resistant. But we do know that in systems like the placental syncytium, there are totally new traits that are acquired by that system, where you are simultaneously having ion channels that are not normally expressed with each other, that are expressed on the cell surface, that enable that placenta to really operate as many different types of organs simultaneously for enabling nutrients and other components to transfer between the embryo and the rest of and the maternal system. So there is lots to be learned in terms of what exactly is happening in this syncytial state. What we're showing is just taking any normal fibroblast and driving it to fuse together does some amazing things that we think actually underlies the jump start to differentiation and the change in gene expression that's happening in these other systems.
Okay. Thank you for your talk. So, as in the case of, let's say, mitosis, where CDK is sort of expected to take over whatever in the cytoplasm in there and make everything mitotic, the question is, in the case of YAP1 or any other type of fusion, is it that there's a dominant signaling or enzymatic pathway that takes over the cytoplasm, like the dominant one takes over the passive one? And like in natural settings, is it like a purposeful fusion that has dedicated signaling, or whether it's just the dominance of one cytoplasm over the other?
That's the big question in the field. In these developmental systems where you have fusogens that are being expressed, they get turned on at a particular time in development, and then you have the cells undergo fusion. What we're showing here is we can recapitulate a lot of stuff that's seen in that complicated system in a very simple system. simply by driving fusion with VSV-G, and we can time it. We're not just looking hours or days after the cell-cell fusion and seeing changes. We're seeing very rapid changes over time. Now, getting to your question about the mitosis, when cells are, if you look at the steady state distribution of a population of cells in terms of interphase versus mitosis, most cells are in interphase relative to mitosis. So when you drive cell-cell fusion, the majority of cells that are fusing with each other are interphase cells. And they immediately put themselves into a differentiated state where they go out of the cell cycle. If, however, you fuse a mitotic cell by chance with an interphase cell, what our data is suggesting is that the mitotic cyclins and other things that are in that cytoplasm of the mitotic cell can be dominant and drive very quickly the incoming cell to go into mitosis. A consequence of that is the system is going to blow itself up because these mitotic nuclei don't know what to do. I mean, they just start pulling. I mean, we saw an example, basically, of a syncytia pulling itself apart as a consequence of a dividing cell. So that might be one function. If it's the case that you have a single mitotic cell fusing with 15 nuclei that are in interphase in a syncytium, that mitotic cell will not dominate. It won't drive all of those other nuclei in. It's only when you have a close to equivalent ratio in terms of the number of interphase versus mitotic nuclei that the mitotic nuclei, the cytoplasm of that mitotic nuclei dominates.
Wow, I think half of the room have questions. And we can stay here forever for the entire night, just asking Jennifer questions, and she'll be here
I'll be happy.
hours and hours.
Please, yeah. We should probably get going. Yeah.
Well, for the sake of time, I'm so sorry to say that. We probably need to move on, but Jennifer's going to be around, and we have a lot of great discussions during beer hours, and tonight and tomorrow and the day after. So
thank you all so much. I really look forward to talking to all of you about this.